Wonderful. Thank you very much. Bonsoir. Um, my great pleasure to talk here. My only sadness is I'm not with you in Paris, but here's hoping for the future. I'd like to talk today about the neurobiology of autism spectrum disorder genes. This is very much an emerging field, but really thinking about the big picture, what does this tell us about what autism is? So let's start off here. I think you've seen this slide already today, and similar ones like it in schizophrenia. This is our effort of where the genes are. And of course, this is work done not by me alone, but by the whole of the autism autism sequencing consortium. Of here, you can see the working group. There's far more people contributing samples. So Joe touched on this, but I'd really like to dive into this more. When we look at these genes, we find that they tend to have a high degree of functional convergence. So doing a very, very simple analysis, looking at the gene, looking at its function, we find that far and away, the majority of them are gene expression regulators. And by this, we mean transcription factors or genes which interact with chromatin, the protein on top of DNA, and a few of them interact with RNA. But all of these group here, there's 58 genes shown in this purple group here. Their main mode of action, we believe, is by changing the expression of other genes. They're essentially part of the regulatory framework which makes our brains work. The second group of genes we see is this neuronal communication group shown here in green. And these are genes which generally are at the synapse, as part of its structure or part of its receptors, or are ion channels allowing the flow of electrochemical gradients to actually make the neurons work. And then there's this group at the bottom, sort of 20 genes. Cytoskeleton has emerged from this, shown in gray. And then there's another group in white where we're looking at autism alone. We don't really see a clear pattern. But if we do this statistically, if we look at gene expression as a gene function using, say, Panther or gene ontology, we find these signals of gene expression regulation, neuronal communication, and cytoskeleton come out. So the question then becomes, well, how do all of these plan together? And what makes autism distinct from, say, the other disorders? So thinking first about this question of other disorders, if we go and look at other disorders where we have equivalent gene lists. So on the left, we have neurodevelopmental delay. This is a genes list provided by the DDD consortium, based in the UK, but also working in Europe. And they recently had a paper, the Kaplanis paper, with 285 genes. In autism, we just saw we've got 102. And then TJ showed you earlier that you've got 10 from the Schema consortium in schizophrenia. Now, if I go and look at the function of these, we find that not only is gene expression regulation the biggest category in autism, that's also the case in neurodevelopmental delay. And it may be the case in schizophrenia. When you've only got 10 genes, it's hard to sort of talk about equivalent numbers, but certainly that signal is present. And secondly, when we look at the neuronal communication group in green, we see that is also present in these. It's a large part of neurodevelopmental delay, big part of autism, and maybe even bigger part of schizophrenia if that trend continues. Similarly, the cytoskeleton pattern is there. And when we look at this scale, when we've got hundreds of genes to work with, we also see a fairly large contribution coming from signaling and a fairly large contribution coming from ubiquitin, which is maybe not a surprise given the Angelman group. So in some ways, this is actually simplified our question when we start thinking about the fundamental question, what is autism and neurodevelopmental delay and schizophrenia? We find that if you look at gene expression regulation and neuronal communication genes, and maybe signaling and cytoskeleton, that actually seems to capture most of the signal we've seen here. And if we go and do equivalent analyses in the common variant space, these are all based on rare variants. In the common variant space, we find the same signals of gene expression regulation and neuronal communication in schizophrenia, where we've got a large data set to work with, and maybe an immune signal as well. So the next question then becomes, well, where and how do these act? So the first thing which we've looked at in the ASC group, and a lot of this is from the ASC paper, if we take those genes and simply ask the question, which bit of the body are they expressed in? Now, we ask this in a slightly more sophisticated way. If we take genes which are not evenly expressed across the entire body, which tissue do they tend to be enriched in? And in black, you can see all of our 102 autism genes. And the answer, maybe unsurprisingly, is brain. Encouraging to know that's the answer, but at least coming at this from a unbiased genetic perspective. But it gets more interesting if you start to split them into the gene expression regulation group and the neuronal communication group. So gene expression regulation group, shown in purple here, while it is slightly enriched in the brain, it's not dramatic. In fact, it's not even significant after you correct for the 53 tissues we tested. In contrast, the neuronal communication genes are very, very specific to the cortex, or very, very much enriched in the cortex. And so here we have the first difference between these two groups. The gene expression regulation genes are ubiquitously expressed generally. They're expressed in almost all tissues, whereas the neuronal communication ones, unsurprisingly from their name, are much, much more cortex and brain specific. If we look at their expression profile over time, again, we start to see a difference here. So we find that the gene expression regulating group, shown in purple here, tends to be expressed prenatally. So what you're seeing here is the gene expression. Every thin line represents one gene. The thick line represents the group together, the composite. And in black, you can see all of them. So generally, the genes which we're interested in the brain are expressed at a higher level in the prenatal period than the postnatal period. This is particularly true for the gene expression regulation group, but actually less true for the neuronal communication, where they tend to rise across prenatal development and then plateau. And so these profiles are really surprisingly distinct. We can put a number onto that by asking, if we ask the question, do they tend to have more expression before birth or after birth? We see this clear profile here where there's a distinction between the neuronal communication and the gene expression regulation, where we see a statistically significant shift of what looks obvious to us in looking at that previous graph. But there's a slight problem in looking at gene enrichment. And so that the way we're looking at there is we're making a big assumption, and that is that the peak of gene expression equals the peak of its activity in causing autism. And actually, we have no guarantee of that. So another way of looking at it is the absolute gene expression. And here we follow a much more simpler logic. If the gene isn't there, then it can't be doing anything. And so on the right, we can see, asking the simple question, at what stage in development do we start to see all of the genes being expressed? And so the top line is probably the simplest one to interpret. And this is asking the question, at what stage do we see the gene expressed in 1% of cells? Now, that doesn't mean it's only 1%, because single cell technologies only pick up a fraction of the transcripts which are present in the cell. But if we take that definition, we see that somewhere around prenatal development, we get to the stage where basically all of the autism genes are expressed in the developing human cortex. And if we take a slightly more conservative threshold, we still find that somewhere around that mid-fetal stage is the point where we start to see most of the genes being expressed. And so going back to the idea of, can we use these observations to understand neurobiology, we see here that there is a hint that prenatal action might be important, but that's not a guarantee. That we see that is different between the gene expression regulators and the neuronal communication. And we see that if we take an absolute point, that we can maybe rule out the early prenatal period, because not all of the genes are expressed there, if we believe that we need all of these genes to be present to tell us when the true autism biology is present. Now, this, I think, is a discussion which has a lot of implications. Putting this very simply, if all of the neurobiology of autism is occurring prenatally, that's going to make it much harder to treat. However, if it's more of a postnatal action, that might have more therapeutic potential. But again, this is speculation because we don't really have any hard data to make these judgments from. So we can use the same data to try and make sense of which cell types are involved in autism. And so here I'm going to rely on some data from Tom Nowakowski and Arnold Krigstein. This is single cell data from the developing human fetal cortex. And every single dot represents one cell taken across multiple samples. With those cells, you can look at the gene expression and put them into clusters. And those clusters, labeled with a C, are basically representative of cell type or cell state. When we look at these, we see distinct clusters related to the glia, for example, microglia on its own over here, whereas we see this group of clusters related to neuronal development. In the sort of pink color, we see cells which are related to excitatory neurons and the excitatory neuron lineage. Up at the top in blue, we see the inhibitory neuron lineage. And generally, up in the top left, this is the area where the cells are developing and dividing, whereas they then mature and differentiate as we go towards the right. So we can take all of these cells and ask the question, where do we see our autism genes expressed? And so when we do that, we find that there is enrichment for the autism genes in both the mature excitatory and the mature inhibitory neurons. And this suggests that both of these neuron types are involved and that that there is relatively little signal coming from glia. In fact, we see basically nothing in microglia or endothelium or the choroid plexus, maybe a hint coming from the oligodendrocyte precursor cells and astrocytes. If we then ask the question, how do the gene expression regulators and the neuronal communication genes interact with this? We find that the same pattern is really present in both of them. We see, let's start with neuronal communication, an enrichment here, particularly in the excitatory neurons, but also some in the inhibitory, but there's a little bit going on over here as well. We actually see a clearer picture in the gene expression regulation genes of that being more specific to the mature excitatory and inhibitory neurons. And this is maybe something of a surprise because remember the gene expression regulators are fairly ubiquitously expressed. It's maybe a slight surprise that they show the true specificity here in cell type. We can take this logic and extend it. And so taking gene lists achieved from rare variants and common variants, we can ask the expression in the developing human brain, where do we see this level of gene expression enrichment? So we just looked in autism, and that would be this area here and this area here, representing those same mature excitatory and inhibitory neurons. I'm going to remove or simplify this to try and bring out the significant and substantial findings. And what we see here is if we look in developmental delay and autism spectrum disorder and epileptic encephalopathies, along with educational attainment, we generally see the same pattern across all of them. And that is distinct from the patterns which we see in the neurodegenerative disorders, which tend to have more of a glial focus. So here's another way of showing that same data. So here I'm showing the development of those different neurons. So we're seeing the inhibitory lineage on the left, the excitatory lineage in the middle, and then glia on the right. And the differentiated mature neurons are at the bottom, the immature developing ones are at the top. And in red here, we're showing the gene enrichment, and we saw that that was particularly strong for these mature and maturing interneurons and excitatory neurons at the bottom. So this is the same image we've just seen. But just like we said that there's a bit of a problem with looking at gene enrichment when we consider the peak of gene expression for timing, the same is true in cell type. Just because the peak of expression, which drives it, is in a certain cell type doesn't guarantee that that is the cell type where the actual effect is taking place. And in the same way as we thought about absolute gene expression, whether the gene is present or not, for time, we can ask the same question for cell type. And so if we do that for cell type, we take our 102 genes, we can ask the question, are they present, which is shown as a red part of the pie chart? Are they not present at all? And then in grey, we have genes which are not present in any of these cell types at all. So that probably represents a limitation of the single cell technology, because when we use bulk RNA-seq data, we can find that these genes are expressed in this tissue. And similarly, when we don't worry about clusters, we can find these genes expressed in some of them. So let's mentally ignore the grey pie chart and put that down to technological limitations. The question then becomes the difference between not being expressed versus being expressed in each cell type. The key difference we see here is down in the excitatory lineage, we see the most absolute expression of these cell types. There's a sort of summation at the bottom of it here of all of the excitatory lineage together. And you can see that large number of these genes are expressed. But even if we take our best lineage, the mature, excitatory neurons, we still find that there are some genes which are not expressed. And because they're in blue, that means that they are expressed in some of the other cell types. And so if we start off with the theory that the true cell type which causes autism needs to express all of the genes which cause autism, then we find that we cannot do that by these developing excitatory neurons alone. In fact, we need to bring in another class of cells to make sense of this. I'm going to show this in a different way. If I pick these two genes, SLC6A1 and DSCAM, we're going to look at the expression pattern of each of those. Now, note that both of these are above the red line, and that red line represents a very stringent level of genome-wide significance. So here, we're not discussing whether these genes are correct or not. These are true findings in autism, and so anything they tell us about cell type is information about cell type rather than casting doubt on their expression or their reality. And so when we look at these two genes, SLC6A1 and DSCAM, we find that their pattern of expression is really quite distinct. So SLC6A1, which is a GABA transporter, is basically specific to inhibitory neurons in the developing brain. We see it a little bit earlier on in some of the developing neurons over here. In contrast, DSCAM is only expressed in excitatory neurons. And so if we want a model of what the autism neurobiology is, the key take home here is it needs to include at least inhibitory neurons and excitatory neurons, because you cannot get to a single cell type which explains all of the genes we find alone. And this builds, I think, strongly on past concepts of excitatory inhibitory balance and the relationship between these. But I think it gives us an important jumping point to go forward. This data is limited to the human cortex. It is possible that sometime in the future, as we start to explore single cell profiles of more brain regions, more stages of development, it may be that we come to a brain region where there is a single cell type which explains all of the autism genes, and therefore that would have a strong evidence of being a nexus point in autism neurobiology. But right now, cortex is the region to beat, and there you need these two cell types to have a consensus view. So that then brings us to the next question. Well, what exactly is the relationship between these two groups of genes? Do you need gene expression regulation and neuronal communication to lead to autism, or will one or the other do? And then in a similar way, do the gene expression regulators regulate the neuronal communication genes leading to autism, or do both of them independently contribute risk to autism without interacting together? Now, the top one seems like the simplest model. Since these are genes which act through a regulatory mechanism, it would make a lot of sense that they act through the neuronal communication genes leading to autism. Now, this is an answer which I think it's fair to say is not definitive, but we're seeing some interesting data which is making us question whether that model is the correct one. So what we did here is, and this was run in large part by Arjament, who's in Turkey, we've collected data on ChIP-seq results. So asking for these gene expression regulatory genes, which genes do they regulate? We collected that from all available data sources. Now, this was a bit of a grab bag. A lot of it was in mice, some of it in rats, tiny bit in humans, and a lot of it was in cell types which we wouldn't generally associate with the brain, for example, fibroblasts or embryonic kidney cells. But seeing as these are ubiquitously expressed, that seems like a reasonable place to start looking. And so at the top, we have all the genes in purple, where we were able to identify some data that they were having a regulatory effect on other genes. So, for example, there's some ChIP-seq data. And the first question we asked is, do they regulate themselves? And the answer there, shown by this p-value here, is yes. So there is feedback within and between these genes. Secondly, do they regulate other gene expression regulators? And again, the answer is yes, shown by this p-value here. We see far more interactions there than we would expect. But if we then go and ask the question, do they regulate neuronal communication genes? The answer is no. We actually see a little bit more we see less than we expect by chance. And similarly with cytoskeleton, we see less than we expect by chance. And so this is some slightly tenuous data that the simple relationship that the gene expression regulatory genes regulate neuronal communication genes doesn't seem to be supported. Now, you might look at this and be thinking, well, hang on a minute. You just told me that these neuronal communication genes are somewhat specific to neurons, and that a lot of this tissue which went into this analysis didn't come from neurons. So maybe that regulatory relationship is just not there if you're looking in the wrong cell type. So we can take one of our top hits. So this is now focusing on CHD8, which is right there at the top of our chart. It's one of the top 3 genes for evidence in autism from exomes. And if we ask the question there, do we see the gene expression genes being targets of CHD8? The answer is a very, very definitive yes. So we expect to see about 5. We actually see about 30. And that is a dramatic difference based on this normal distribution. If we do the same for the neuronal communication genes, and this is now in neuroprogenitor cells, we find that there is actually less than we would expect by chance, supporting the idea that the simple relationship that neuronal communication genes are the targets of the gene expression regulation genes doesn't seem to pan out. Now, again, you could still say this isn't quite the perfect experiment. Maybe we need to be doing this in excitatory neurons, and there we just don't have the data. But at the first glance of this, that simple relationship there doesn't, at the moment, have data to support it. So then what happens? What is the relationship between these? Now, I don't have a definitive answer to that, but if we go and look at SCN2A, we can come at this problem in another way. So SCN2A is a gene which we pick because it's the top of the evidence from the exome data. And also is a very, very intriguing gene. It's intriguing because it leads to 3 distinct phenotypes. So if you look at children with variants, rare variants in SCN2A, you find 3 different phenotypes. The first one is infantile epileptic encephalopathy. These are very severe early onset seizures leading to long-term profound learning disabilities. You then find another group of children who have benign infantile seizures. These have the same early onset seizures, so seizures before the first year of life, but then they go on to have the seizures resolve and no long-term problems. And then finally, there's autism developmental delay, where there are no seizures in the first year of life, sometimes seizures later, but they have the classic impaired social communication and learning disabilities that characterize autism and developmental delay. Looking at the function of SCN2A, which is a voltage-gated sodium channel, we can find that there's a nice clear explanation for these different phenotypes. If you have a gain-of-function mutation, which is severe, that leads to infantile epileptic encephalopathy. A mild gain of function leads to benign infantile seizures, and then a loss of function leads to autism and or intellectual disability or developmental delay. So this gives us a nice starting point to go and look in a mouse to start asking the question, well, what's actually happening at the level of the neuron? And because it's a sodium channel, we also know where to go and look. We want to go and look at the sort of biochemical, electrophysiological properties of the sodium channel, of the neuron. So looking here with patch clamping in wild-type mice versus heterozygous loss-of-function SCN2A mice, we find that early in development, we have a reduction in the excitability of neurons. And that makes sense, seeing if you have impaired conductance of SCN2A, which is responsible for creating action potentials. But SCN2A undergoes a profound shift in the middle of development. Early on in development, SCN2A is the only sodium channel creating action potentials in excitatory neurons. And so it's responsible for action potentials being fired onto the next neuron, but also back propagation, which is the process by which the action potential goes back to the dendrites and essentially trains the synapses, which led to the signal in the first place. But around 1 year of age, that subcellular location changes as SCN2A is replaced by SCN8A at the distal axon initial segment. And that means that SCN8A is now responsible for initiating action potentials and forward propagation, and SCN2A is relegated to only back propagation. And around the same time, which is about 1 year in a human, there is also a splicing change going from the neonatal isoform in exon 5, which is less excitable, to the adult isoform, which leads to a slightly more sensitive neuron. So essentially, a profound shift happens around the age of 1 year in SCN2A, having a big impact on what the neuron is doing in respect to SCN2A. And so that means that if you go and look at our reduction in excitability, which we saw in these mice, it goes away. But in contrast, if we ask the question about back propagation, about how fast is the action potential fired back up the dendritic tree to go and train the synapses, we find there is an emerging deficit which comes around that switchover about 1 year of age in humans. And so we see this emerging loss of the action potential being fired up the dendritic tree. And that leads to immature synapses. And so without that signal telling them that you successfully fired, well done, get stronger, looking at the AMPA to NMDA ratio, the synapses remain in an immature state because they've not had that signal come to them. And so we find, looking at SCN2A, we get to a very, very similar question, whereas we looked at the cell types and the timing of these genes all together. So the first one is, when does the true pathology of autism occur? Is it this early developmental phenotype, which presumably leads to something permanent, which we're not able to assay at the moment? Or is it this learning phenotype, which happens later on, this lack of maturity, which represents the process of learning in the synapse? Or is it both? And this is very similar to the sort of gene expression regulating profile versus the neuronal communication profile of the genes. And then over here, we have this essentially learning deficit induced by this loss of sodium channels. Could we see something similar coming from the gene expression regulating genes? If they're not directly regulating SCN2A, maybe they're involved in feedback in the very, very process of learning, leading to convergence at the synapse in their ability to synthesize information. All right, so that's my summary of where we've got to in terms of the sort of the key corner pieces of the neurobiology of autism. We've talked about gene discovery, and I think your sessions today have highlighted the importance of that, of driving forward these kind of efforts. The good news is we see profound functional convergence. We've seen these gene expression regulation signal and neuronal communication gene signal, and maybe weaker ones coming from cytoskeleton, ubiquitin, and signaling. But these are kind of the corner pieces. If we can just work out what those bits are doing together, hopefully the entire picture will start to emerge. And then finally, we've seen cell type convergence. We see that these two major sets here are acting on both excitatory and inhibitory neurons, but that a model which includes only one of those doesn't really make sense in the picture. We need both neuron types, leading to the potential of autism being a EI excitatory inhibitory balance or a pan-neuronal learning deficit based on current knowledge. And I'd like to end by thanking all the wonderful people, both at UCSF and in my lab, and collaborators who work with me and have been a key part of this journey, particularly highlighting the Autism Sequencing Consortium, which is the top 5 people over there.